Hello class, we are topic 2, so we are going to be going over fermentation and catabolism of organic compounds other than glucose. And I am Mikey Alvarez. My first slide is going to be on fermentation. So what is fermentation? I'm sure we've all learned about cellular respiration, and so it might ring a few bells, but fermentation is the anaerobic process which organisms use to produce NAD+, which helps keep the process of glycolysis going. Why is it important, you may ask? It's necessary because it produces ATP when oxygen is scarce. And like I said previously, we need ATP to stay alive. Fermentation produces few ATP, so we couldn't live off that alone, and there are many organisms that can. For instance, like bacteria, archaea, yeast, and even our own muscle cells.
Okay, like Mikey was saying, fermentation and cellular respiration are very similar, so we're going to go over some key differences. Fermentation is the anaerobic process that generates ATP without oxygen, whereas cellular respiration is the aerobic process that does use oxygen. Fermentation only generates 2 ATP per glucose, whereas cellular respiration creates 36 to 38. So cellular respiration is a lot more efficient, but fermentation is still helpful when there's no oxygen available. Fermentation uses organic molecules like pyruvate as the final electron acceptor, because there's no oxygen to use as a final electron acceptor like cellular respiration would. But they both create ATP, NAD+, they both begin with glycolysis, and they both occur in the cytoplasm.
Alright, so why use fermentation if it only produces few little ATP? In this case, I'm going to be talking about the smaller organisms. So for instance, some organisms use different electron acceptors to survive, while others don't have the electron transport chain, and that makes it a little difficult for them to complete glycolysis and produce ATP. Some organisms that use fermentation are E. coli. They can switch between cellular respiration and fermentation. And another is streptococcal pneumonia, which must use fermentation because it doesn't have the electron transport chain.
Hey guys, I'm Angel. So right now we're going to talk about the 2 major types of fermentation. We got the lactic acid pathway and the ethanol pathway. There's some more that I'm going to cover in another slide, but these are the 2 most common. So in lactic acid fermentation, this is the one that we learned in anatomy. Basically, that burn that you feel when you're doing heavy exercise like sprinting, it's the buildup of lactic acid, which comes from the electrons being dropped off of pyruvate because there's not enough oxygen being made, so your cells can't grab the oxygen. And the NADH plus gets oxidized to become NAD plus, and then you're able to keep glycolysis going. But that, as a byproduct, you create lactate, better known as lactic acid, which is that burn that you feel in your muscles. And the lactic acid gets processed in the liver, and then the process can repeat. And in ethanol fermentation, NADH plus donates its electrons to a molecule called acetaldehyde, which comes from pyruvate, and produces CO2 as the product.
Alright, so how does fermentation affect your medical and just regular life in general? So in medicine, fermentation can be used as a diagnostic tool to differentiate bacteria, and it can also be used to make essential medications like probiotics, antibiotics, vaccines, insulin, so on and so forth. Also good for human health. It's good for your gut relationship with your brain. So for instance, if there's an imbalance of microbes in your gut, that can lead to many problems. Throughout life, it helps in many different things, helps preserve many foods, for instance, many vegetables, beers, and so on and so forth. And it also helps provide more nutritional values, and like I said previously, it aids in gut health. It also offers new textures and flavors to many foods.
So on this slide, we have different end products of fermentation. So the 2 main pathways that I talked about, lactic acid, ethanol, but there's also 4 more for the products of some of those. And then this is commonly known as ethanol poisoning. So, but yeah.
Okay, so other than making food, there's a lot of industrial applications to fermentation as well. One of the big ones is biofuel, which you can ferment corn and sugarcane to create ethanol, which is a good ethical replacement to gasoline. Over 1.5 billion gallons of ethanol biofuel were produced in the US 2 years ago. Another option is bioplastics, which certain bacteria can ferment sugars into biodegradable plastics, which can be used to make water bottles and lots of important things. And then as far as waste treatment, fermentation can be put into the sewage pipelines or used in the pipelines to break down the organic matter in there without the need for oxygen in the pipeline itself.
Catabolism of organic compounds. Catabolism is the way microorganisms get ATP and small molecules that play a role in anabolism. Glucose is the most common energy source for many of these microorganisms, but when this is scarce, they can catabolize other organic compounds such as lipids and proteins. An exception to this would be the bacteria chemolithotrophic, because they cannot break down glucose. This flexibility ensures that they can thrive in different environments, such as soil and our guts. Some microbes live in environments that are rich in organic matter, and they heavily depend on protein, but it's really important to note that only microorganisms that produce extracellular proteins can catabolize protein. Protein, apart from energy, also provides nitrogen and carbon. And as a summary, protein is hydrolyzed into amino acids, which are then denaminated. Basically, the amino acids are broken into its two components, and then all you are left with is this carbon skeleton and the ammonia, which is a waste product, and the carbon skeleton is the actual fuel source that they use to get their ATP. And two examples of microorganisms that can't break down protein would be the fungi Penicillium and the bacteria Bacillus. And these are just two pictures of those.
Hi, my name is Josie, and I'm going to be talking about the processes of organic compounds. The first one that we're going to go into is protein. So there are 4 steps to protein catabolism. The first one is for enzymes to break up the protein into individual amino acids. After the nitrogen has been removed from the amino acid group, the amino acid either makes pyruvate, acetyl-CoA, or goes into the TCA cycle. The next process that I'm going to talk about is catabolism of a lipid. When a triglyceride is broken up, it is broken up into 1 glycerol molecule and 3 fatty acid chains. Once the glycerol is by itself, it enters a cell where the enzyme glycerol kinase performs phosphorylation. G3P is oxidized into DHP by the enzyme glycerol 3-phosphate dehydrogenase. DHP is converted back to G3P to begin glycolysis. The other product of lipid catabolism is the 3 fatty acid chain. The fatty acid gets oxidized until it becomes acetyl-CoA units, which are then used in the electron transport cycle to create ATP.
Perfect.
The third process of catabolism that I'll be talking about is catabolism of nucleic acids. So there are 5 steps to nucleic acid catabolism. The first one is the breakdown of the acid to nucleotides. Nucleotides are DNA and RNA are hydrolyzed by nucleases into ATP, UTP, GTP, CTP, and TTP, which are all triphosphates. The nucleotides are then broken down into nucleosides and phosphates, which means that the phosphates are taken out of the nucleases. What's left is the phosphates groups and the nucleosides, which are adenosine, guanosine, cytidine, thymidine, and uridine. Okay, the third step is nucleosides are then broken down into their nitrogenous bases and sugar, which basically means the sugar is taken out of the nucleotide to just leave the base. There are 2 different categories of the bases: purine bases, which are adenine and guanine, or the pyrimidine bases, which are cytosine, uracil, and thymine. Sugars are deoxyribose, which is used in DNA, and the ribose, which is the sugars in the RNA. The 4th step is that sugars can then enter glycolysis, which means that they're breaking down for energy. Purine bases are then converted to uric acid and excreted, and the pyrimidine catabolism means that the product will be reused in metabolic pathways.
So why catabolism? As stated earlier, catabolism is the way microorganisms get ATP, which is energy, in smaller molecules that play a role in anabolism. This takes place through the breaking down of complex molecules in lipids, proteins, carbs, nucleic acids, and other stuff. This is known as catabolic reactions. There are 4 main catabolic pathways that are used: glycolysis, which is the breaking down of glucose into pyruvate; the citric acid cycle, also known as the Krebs cycle, which is the oxidation of acetylcholine to CO2; and then beta oxidation, which is the breakdown of fatty acids into acetylcholine; and proteolysis, which is the breakdown of proteins into smaller amino acids. The first catabolic reaction is glycolysis, which is the process of breaking down glucose into pyruvate. This happens in the cell cytoplasm and doesn't need any oxygen. It's also known as being anaerobic. Glycolysis also makes a small amount of ATP. The process itself makes 4 total, but 2 are used during the process, so it only has a net total of 2 ATP. And it also makes 2 NADH. The second pathway is the citric acid cycle, also known as the Krebs cycle. This happens in the mitochondria and uses something called acetylcholine, which comes from the carbs, fats, and proteins. In this cycle, acetylcholine gets broken down into the carbon dioxide, which is an aerobic reaction. The third pathway is beta oxidation, which breaks down fatty acids into more acetylcholine. That acetylcholine then goes into the Krebs cycle to make more energy. This process is really important when the body is low on carbs, like during exercise, or if someone hasn't eaten in a while, such as when they're fasting. The last pathway is proteolysis, which is when the body breaks down proteins into amino acids. These amino acids can be turned into other useful things like pyruvate or acetylcholine, which can be used for energy just like before.
And then I'd like to incorporate a little bit of a Christian worldview into the future of our careers as healthcare workers. It's important to know that why we're here, to help others, to help those that can't help themselves, those that are in need of help. And it's important to be compassionate. That's something that can't be taught in class. So just be mindful of that. And whenever you are in your job and you're working in the clinic or wherever, in the hospital, when you do as much as you can medically, when you do all that you can medically, if you really care about your patients, go home, pray for them, and let God handle the rest. And then I added 2 Bible verses. Galatians 6:2 says, "Carry each other's burdens, and in this way you will fulfill the law of Christ." And that means all type of burdens: emotional, mental, physical. In this case, physical. Praise God. Oh, and then these are the references. Thank you so much for watching our presentation. It was real. Thank you guys.